grade 12 class. Today we're going to be doing life science. The first topic, which is DNA. So, DNA, the code of life. DNA, the code of life. So, before we start, I want you to do not forget to like, subscribe, and share this video to others. So, and firstly, we must look at our exam guideline in order for us to know what they expect us to know in DNA, code of life. So let's go to our exam guideline, which is for 2021. You should have this guideline whenever you're studying so that you can study relevant information. So you can see, firstly, code of life, DNA code of life. It is on paper 2 of life science, grade 12. So let's see the weighting and the marks. It weighs 18%. It weighs 18% and has 27 marks. I hope you cannot afford to lose such marks. These are easy marks. So let's get into it. So DNA code of life introduction. We must firstly do revision on the structure of a cell with emphasis on the ribosome, cytoplasm, and other parts in the nucleus. Two types of nucleic acid. When they say nucleic acid, they are talking about DNA and RNA. We'll go deeper into them. Do not worry, do not panic now. So, and also you must look at the nucleic acid consists of the nucleotides. I know this is a new concept, but we'll get right into them. So let's go into our introduction. So I want us to actually know the position before we get into the structure of DNA. Let's go back to our exam guideline and see. In return, you did structure of a cell. You know the ribosome. What is a ribosome? You know the organelle in the nucleus, which is cytoplasm. You know the nucleus, the type of for the type of nucleotides. I don't think you have prior knowledge about it, but these are the nucleic acids which are found in the nucleus. We'll get right to them. So let's see. They say we must know the location, structure, location, structure, and the function of DNA. I know you're asking yourself, what is the meaning of DNA? Deoxyribosome nucleic acid. Deoxyribosome nucleic acid. Deoxy means that it does not have what? Oxygen. Deoxyribose, sugar. Deoxyribose sugar. It's deoxyribose sugar. So let's go. They say the location you must present in the nucleus. Make up gene on the chromosome. Present in the mitochondria. These are the location. Okay, firstly, one thing I like about this exam guideline, these are types of questions that they the usual ask. They may say, what is other location of DNA except in the nucleus? You will say that in the mitochondria. You say that it is present in the chloroplast. So I want us to go into the structure of a DNA, the natural shape of a DNA molecule. It is double helix. They usually ask these questions, especially on one with term, biological terms. I mean, sorry. Each strand made up of sequence of the DNA nucleotides. So this is the summary. Actually, this is a summary of what we are going to be doing in today's lesson. Three components of DNA. So this one will get right into them. So let's focus on the position. Position, it actually means the location. Position in the cell. It is found. Let's look deep into our cell. Uh, it is found double helix. It is found in the cell firstly. If this are mitochondrial, we can find DNA in mitochondrial. This is a nucleus we can find, and in the cytoplasm. Cytoplasm is like a fluid-like gel that supports the organelles in the cells. Yeah, so we must know that the DNA comes from chromosome. When DNA shrinks, we call this thing a helix. This shape helix, double helix, and when it shrinks, it forms what? Histones. After histones, it rolls until it forms what? Chromosome. So we get right them, we get deep into them. So in the nucleus of almost every cell in your body's collection of DNA needed to make you is true. DNA is the nucleus. DNA in the nucleus is grouped into 23 sets of chromosomes, which are called genome. Yeah, this one is another concept in meiosis. We'll do it in meiosis. Each chromosome, the DNA is grouped into the gene. Okay. Instruction to make your whole body and keep working in DNA. Genetic code, okay. We have dealt with the structure of the DNA, I mean the location of the DNA, where it's found. We know in the nucleus. In the mitochondria, and in the cytoplasm. No, so let's go into the structure of a DNA. DNA consists of two molecules that are arranged into ladder-like structure, which are called double helix. This may come in one with term. A molecule of DNA is made up of million tiny of subunits called nucleotides. I want to show you what is nucleotide. Actually, nucleotides are building blocks of DNA, nucleic acid. When I say DNA and the nucleic acid is the same thing. So nucleotides let's see the structure of the nucleotide. Actually, the DNA that you see here, it is formed by these structures, by these nucleotides. Many or millions of nucleotides forms DNA. So DNA consists of many nucleotides. The nucleotides are made of phosphate, phosphate group, pentose. This pentose sugar is a glucose and also nitrogen base. And we'll dig deeper into this structure. Nitrogenous bases, which are your cytosine, guanine, and thymine, and adenine. So nucleotides, the phosphate and the sugar form the backbone of the DNA. It's true molecule, whereas the base of the form the rank. The ranks means the ladder-like structure. Four types of nitrogenous. These nitrogenous are a complement to each other. They match in order to make double helix, double-stranded. Adenine matches with thymine. Cytosine matches with guanine. This is a tip to remember. Does with guanine, does with cytosine, okay? Then nucleotides, each base will only bond with one other specific base. Adenine, A, and thymine form a base pair. Guanine and cytosine forms a base, also forms a base pair. Then, because of complementary base pairing order, one structure determinant of the bases in the other strand. Sorry. So you see those nucleotides. These are your nucleotides. These are your nucleotides. They form what? They form that structure which is DNA. So DNA structure is a gene section that code for protein. A gene is a section. Okay, you must know the definition of a gene. It codes for proteins. Each unique gene has a unique sequence base. This unique sequence base codes production of a unique protein. And this protein combination of protein gives us unique phenotype. This is you would do this term in genetics. So we have process actually, which is DNA replication. DNA replication simply means that DNA makes its replica, means it's identical gene, identical DNA. It forms an identical DNA. So I want us to go into to look this some guideline. Since like we've already covered, we must know the structure. We dealt with it. It is double helix. The structure's double helix, each strand made up of sequence of DNA nucleotides. We've seen it. Three component of DNA nucleotide, heterogeneous base linked by weak hydrogen bond. You must know that the only force that keeps the two strands together. This is your weak hydrogen bond. This is your weak hydrogen bond. It is this one here. This is your weak hydrogen bond. It keeps the two strands together. Four nitrogenous base. We have four nitrogenous base. We've seen them. Adenine matches with thymine, and guanine matches with cytosine. Base pairing occurs AT, CT, phosphate portion. Stick diagram of DNA illustrate function of a DNA. You must also know this key bullets here. The function of a DNA. sorry. Function of a DNA, and also DNA. Each function makes up a gene which carries hereditary information. DNA carries hereditary information simply because when they are going to, let's say, there is an investigation about the paternal test, the father of the child, they use DNA to check hereditary information. DNA contains coded information for protein synthesis. We'll do this protein synthesis in our next lesson. So we have processes in DNA, which is DNA replication and protein synthesis. And these are the processes. Do not panic, you know these things. DNA replication, the process. You must know when in the cell cycle it takes place. It takes place during interphase. Where in the cell it takes place in the nucleus. How DNA replication take place? It takes place just because of the enzymes which are called polymerase, but here in your exam guideline you don't need to know them. Significance of DNA replication, the importance of DNA replication. Yeah, so this is what you must know. Yeah, this is what I was talking about, DNA replication. You see how the single strand form from this thread, it's separated into two, then it forms another strand which they doubled. So this is the importance of the DNA replication, why it is important in human body cell to double the genetic materials in the daughter cells. So you must know this process. It starts with DNA double helix unwind. It started in this way as double helix, so now it unwinds. It becomes a ladder. After it becomes a ladder, weak hydrogen bond between nitrogenous base are broken and DNA strands separates. They do what? the weak hydrogen bond, it separates. Then they do what? The strands start to separate or unzip. In other words, so each original strand serves as a template. You can say that both strands serves as a template, which is complementary, which its complement is built. Or maybe you can say a weak hydrogen bond to unzip the two strands, and the two strands separate. Both strands serves as a template to form complementary DNA molecule using free nucleotides floating in the nucleoplasm. So, and you can say that free nucleotides, this is what I was talking about. Free nucleotides build DNA strands on each original, and they are complementary. Yeah, you can add that they are complementary to the original strands. This results in two identical DNA molecules. Each molecule consists of one origin strand. Then you are finished with the DNA replication. The importance, it is important in particularly for cell division. It allows each chromosome to be copied onto each new identical daughter cell produced contain the same number of chromosomes. I know you may not understand these concepts right now, but until you do meiosis and genetics, you will have deeper understanding into this previous concept. When you say, okay, this is why they said about the chromosomes, it is important in mitosis to to actually double the genetical material. So in our next segment, we will be dealing with the RNA, also DNA profiling and protein synthesis. Thereafter, the DNA code of life, we wrap it out. Thank you for watching this far. Please subscribe, share, and like this video.